Assalamu alaikum everyone, this is Abdullah Khalil and this is the platform where you will get to learn O-level, A-level and IGCSE biology of different UK boards. Now we are doing the first chapter of A-level biology, which is cell structure, and up till now in the previous video we have done microscopy, basic comparison of prokaryotic and eukaryotic cell. We have done the use of eyepiece, graticule and stage micrometer, the conversion of units. And in today's class, we are going to study about the cell structures and organelles. The first structure that we are going to discuss is the cell wall. Now, cell wall is the outermost structure of the plant cell. It is made up of cellulose. In animal cell, it is absent. In fungi, it is present and it is made up of chitin. In bacterial cell, it is present and it is made up of peptidoglycan. Now, we are going to mainly discuss the cell wall of plant cell, which is made up of cellulose. So cellulose microfibrils, there are small microfibrils of cellulose which are making the cell wall. This is the basic structure. Then we have other structures to keep this cellulose microfibrils intact and stronger. We have pectin, we have hemicellulose, and in some cells we have lignin, like for example in xylem vessels. Now cell wall is really important because it gives the plant cell the shape and the structure and the strength. Now here comes a question. If we place a plant cell, let's say a palisade mesophyll cell and an animal cell like red blood cell in distilled water, what is going to happen? Now animal cell, because it does not have the cell wall, it is going to absorb water and it will burst. At the same time, the plant cell will also absorb water, but there is the cell wall to support its structure, to give it the needed strength. So the animal cell will burst, the plant cell will become turgid. This turgidity, the losing of water or the gaining of water, is really important for the plant. This turgidity gives the plant cell the ability to support the structure of the entire plant. Now, when the cell is made during cell division, initially it has the primary cell wall. Then there are deposits of another layer, which is then called the secondary cell wall. Another thing that you have to keep in mind about the cell wall is that it is fully permeable. That means it does not control the substances that are moving through it, whether from the outside to the inside environment or from the inside to the outside environment. So it is fully permeable. That means it allows every substance to pass through. On the other end, now this is what this cell wall looks like. We have the cellulose microfibrils here, we have the hemicellulose here, and we have the pectin in gray here. Okay. And this is lignin deposits like in xylem cells. The next structure, which is very next to the cell wall in plant cells and which is the outermost layer in animal cell, is the cell membrane, also called plasma membrane, also called cell surface membrane. It is made up of phospholipids and we call it the phospholipid bilayer. When you look at it under the electron microscope, you will see that there are two layers of phospholipids, and this membrane actually controls the flow of substances across it. So this is partially permeable membrane. That means it allows only specific substances to pass through it. Now cell membrane, we are going to discuss plasmodesmata later. Cell membrane looks like this. Now these are two neighboring cells and this is the cell membrane of one cell. You can see the double layer, the bilayer as we call it, and this is the cell membrane of the other cell. In between these two cells, we have the intercellular space. Okay. We have an entire chapter dedicated to understanding the structure of cell membrane. So we are going to discuss it later. And then you will be able to understand how it controls the flow of substances, the movement of substances across it. The next structure is plasmodesmata, simply saying it is the connection between the two neighboring plant cells. The plasmodesmata are small pores which enables the plant cell to transfer substances from one cell to another. This is how the substances pass within the plants. Now moving on, we have microvilli, characteristic of a few animal cells. Microvilli are finger-like projections, as we have in villi, which is present in the small intestine, the third part of small intestine. Intestine is divided into three parts. We are talking about the digestive system now. So the first part of small intestine is duodenum, which is right after the stomach. Then we have jejunum, then we have ileum. In the ileum, we have these finger-like projections. This is how the structure looks like. And these structures are richly supplied with blood capillaries. As you can see, these are richly supplied with blood capillaries. In ileum, their function is to absorb the digested food. The food, when goes to the ileum, it has already passed the mouth, the stomach, the duodenum. So it has already been through the different parts, through the enzymes, and it has been digested already. Now this food will be absorbed in the bloodstream. Similarly, we have small microvilli present in the surface layering of trachea and in the digestive system, as we have already discussed. So the ones that are present in the trachea, their function is to wipe off the mucus. Mucus is a thick, viscous liquid that is present in the trachea to trap the dust particles during inhalation. So these are microvilli. Then we have vacuoles. Now, sometimes the students say that there are no vacuoles present in the animal cell, while there is one vacuole present in the plant cell. This is not entirely true, because there could be sometimes small temporary vacuoles present in the animal cell as well. But this is how the plant cell vacuole would look like. This is very large. This covers the major part of the plant cell, due to which all the other organelles, as you can see in the diagram, are pressed against the wall, the membrane of the cell. Now the vacuole, this is called the permanent sap vacuole. Sap vacuole because it has the sap, a fluid inside it. And the most important function of the sap vacuole is to control or to maintain the osmoregularity of the cell. As I have said, that the losing and gaining of water for the plant is very important, because sometimes the turgidity of the plant cell supports the structure of the entire plant. So it is really important for the cell to become turgid sometimes and to become flaccid sometimes. If you know about the guard cells, you know that the guard cells become turgid to open the stomata, and they become flaccid to close the stomata. So turgidity and flaccidity of the plant cell is very important. Osmoregulation maintains the turgidity. It instructs when the plant cell has to become turgid and when it has to become flaccid. It also stores the food, and it can also act as lysosomes. It has enzymes in it. Then we have the nucleus, which is like the brain of the cell. Now, as we say brain of the cell, what do I mean by that? The brain controls all the activities of your body. Similarly, the nucleus contains or controls all the activities of the cell. How does it control all the activities of the cell? Now, let me just elaborate. Nucleus has DNA. DNA has genes. Genes are going to code for a polypeptide. By definition, genes are the substances. Genes are the small fragments of DNA that code for a polypeptide, a specific polypeptide. Nucleus has DNA. DNA has genes. Genes codes for polypeptide. Enzymes are also proteins made up of polypeptides, coded by genes, present in the nucleus. So that means all the enzymes are also made by the nucleus, which control the activities, the reactions taking place. All the reactions that we have are enzyme controlled reactions. So nucleus has the genes which code for the enzymes, which make the enzymes. Enzymes control all the activities, the reactions. That's why we say that nucleus control all the activities of the cell. Secondly, when we look at the structure of nucleus, this is what the nucleus would look like. It has double membrane, nuclear membrane, which is called the envelope as well. The envelope of the nucleus, the nuclear envelope of the nuclear membrane. Inside, it has a fluid which is called nucleoplasm. Then there are smaller structures which are called nucleoli, which could be more than one. If there is just one, it would be called nucleolus. Then the most important one, the DNA, which is present in the form of chromosomes. If we look at the prokaryotic DNA, it's just the DNA. We call it the naked DNA. But in eukaryotic cells, in eukaryotic nuclei, the DNA is basically in the form of chromosomes. That means DNA integrated with the proteins called histones. You would, in the future, you would look at the structure of DNA, and you will see how the chromosomes are made up with histone proteins and the DNA wrapped around them. But for now, just remember that nucleus controls all the activities. If you look closely, you will see that there are small pores present. This is so the substances can move in or out of the nucleus, only the specific substances. This is all the details that we have just discussed. Then we have endoplasmic reticulum. Now, let's go back to the structure of the nucleus. When you look outside the nucleus in the cytoplasm, you see these channels. Okay, these channels are actually endoplasmic reticulum, and these ones specifically are rough endoplasmic reticulum. We have two different types of endoplasmic reticulum. We have the rough endoplasmic reticulum, so called because it has small granular structures attached to its surface. These small granular structures are called ribosomes, which are the protein factory. Let's keep it here, which I will pick it up later. Now, these small granular structures or the ribosomes are the protein factory. They make all the proteins. Gene codes for the protein. Ribosome synthesizes the protein. Okay, so you should not be confused about these two. Sometimes the students consider that the genes are the one making the proteins. No, they code for the proteins. They provide the code. That means they instruct in which sequence the protein should be made. But ribosomes are the ones that actually make the protein. So ribosomes are found attached to these rough endoplasmic reticulum, and rough endoplasmic reticulum are involved in the synthesis and modification of protein. The other one is smooth endoplasmic reticulum, so called because it doesn't have the ribosomes attached to its surface. And smooth endoplasmic reticulum is present on one side in the cytoplasm, and their function is entirely different. This is how rough and smooth endoplasmic reticulum would look like if they're placed side by side. Smooth endoplasmic reticulum are the channels that basically make steroids, lipid-based hormones like estrogen in females and testosterone in males. When you look at rough endoplasmic reticulum or smooth endoplasmic reticulum under the electron microscope, this is how they will appear. This is the electron micrograph of rough endoplasmic reticulum. You can see ribosomes attached to the surface when you look closely. And this is a smooth endoplasmic reticulum. There are no ribosomes, entirely different functions. Then logically we have ribosomes. The smallest structure within the cell that we are going to study is ribosomes. This is 25 nanometers in size for eukaryotic cells. For prokaryotic cells, this is 20 nanometer in size. The prokaryotic ribosomes are called 70S ribosomes. This is because of their sedimentation rate. 70S ribosomes and eukaryotic ribosomes are called 80S ribosomes. This is what the structure would look like. They have the larger subunit, smaller subunit, and in between we have the instruction for protein, which is given by the gene. This is in the form of RNA. This is called messenger RNA or mRNA for short. This comes in between the smaller and larger subunit of the ribosome, and this gives instructions step by step of the sequence of protein. Now, if you know something about proteins, proteins are very specific to their function. Just look at enzymes. They are very specific to their substrate because they have a very specific shape. And that shape is defined by the sequence of amino acids in that protein. And the sequence of amino acid is instructed by the mRNA, which in turn has been instructed by the gene. Now, I know I'm making it a bit complicated. Let me simplify it for you. So the shape of protein depends upon the sequence of amino acid. According to that sequence, the protein will fold and make a specific 3D structure. And that sequence is coded by the gene. That sequence is instructed by the gene. So we can say that gene basically controls the shape of protein because it controls the sequence of amino acids. It controls the shape of the protein, hence the specific functionality. Now, just remember from our O-levels and IGCSE knowledge, we have studied that enzymes can become denatured when they are at very high temperature, for example. So, what do you mean by enzyme becoming denatured? That enzyme loses its shape. And when the enzyme loses its specific shape, it is no longer able to bond with its substrate. The active site has lost its shape. So the substrate will not fit into the active site. Hence, the enzyme will not be able to perform its function. So the functionality of protein depends upon the 3D structure of protein. Moving forward, we have Golgi apparatus. One more thing, ribosomes are made up of RNA and protein. Golgi apparatus has the function of collecting, sorting, packing, and transferring. I'm going to repeat myself. They collect, sort, pack, and transfer substances, materials that are made inside the cell to their destination. Now, let's say lipids or proteins that are manufactured within the cells, sometimes they have to be modified. So, Golgi apparatus would modify these structures. Let's say the lipid that has been made can be converted into glycolipid. The protein that is made in the cell can be converted into glycoprotein. That means a carbohydrate chain is attached. So a simple protein with a carbohydrate chain attached to it becomes the glycoprotein. Glycoprotein can be made by the Golgi apparatus. Now, there is something amazing about the Golgi apparatus, that it is being made on one end, and on the other end it is being broken off in the form of vesicles. It makes vesicles in which the substances are packed and then they are transported to their destination, which could be within the cell or outside the cell. Okay, they have other functions, making glycoproteins, glycolipids, and they release mucin. We have talked about the mucus in the trachea. Mucus has an important structure which is mucin. Mucin is part of mucus. So mucin is released by the Golgi apparatus of those cells which are releasing mucus. Next up, we have mitochondria. This little organelle is enabling me to speak to you, because to speak to you, to do this hand gestures, I need energy, and mitochondria is the organelle that makes the energy. Simply, we can say that mitochondria controls, or mitochondria is the organelle which provides energy. That energy is stored in ATP, and this energy is provided or made during cellular respiration. That means within this mitochondria, the food is being oxidized and the energy is being released, and that energy is being stored in the form of ATP. So ATP can be broken off easily. It releases energy, and then we use that energy to speak or to do the hand gestures. That means muscular contraction, which could be blinking of eye. It also is needed to keep the body warm for protein synthesis, for making different substances, for the reactions that are taking place in our body. Another important thing about the mitochondria is there is a theory that mitochondria and chloroplast, they were ancient living bacteria which basically started living with the eukaryotic cells and in a mutual relationship. That means they both provided benefit to each other, mutually beneficial relationship. Mitochondria gave energy to the cell. Cell in return gave shelter and food to this bacteria. With evolution, these mitochondria and chloroplasts, they become essential part of the cell. Now we call them the organelles. Mitochondria are essential organelles of the cells. What is the evidence of this theory? Well, chloroplasts, they have their own plasmids. They produce their own proteins with the help of genes in their plasmids, and their structure is similar to the ancient living bacteria, plus the ribosomes that they have are exactly like the prokaryotic ribosomes. That means the 20 nanometer in size ribosomes, the 70S ribosomes. So because of these evidences, we consider that the mitochondria and chloroplast were actually ancient living bacteria. Next structure that we are going to study is logically chloroplast, only present in plant cell, and I can say that chloroplast is the organelle on which the entire life depends. The life on earth depends on this structure, because chloroplast is essential for photosynthesis. Without chloroplast, there is no photosynthesis. There is no photosynthesis, there are no plants. And if there are no plants, there are no animals. So all the life on earth depends on this structure. And this chloroplast is comparatively larger in size than the mitochondria. It is also double membrane. It has a fluid. Mitochondria also had a fluid which was called matrix or mitochondrial matrix. Within the mitochondria, you can see that there are two membranes and the internal membrane is folded. The outer membrane is smooth, the internal membrane is folded to provide higher surface area. You will study the process of respiration in detail in your A2. The first chapter of your A2 will be about respiration. In chloroplast, we have membrane extensions inside. These are called lamella, or plural is lamellae. Not written here, so I'm just going to write it, lamellae. Then we have thylakoid, which are stacks of small granular structures. These are called grana, and the stacks are called thylakoid. The fluid that we have here is called stroma, and they have small pigments which trap the sunlight. The primary pigment is chlorophyll, but there are other pigments as well. They trap the sunlight, and they are arranged in a specific cone-shaped structure which is called photosystem. Then we have centrioles and MTOC. This is the structure of centrioles. Total of 9 triplets of microtubules. 1, 2, 3, 4, 5, 6, 7, 8, 9. This centriole is present just outside. Actually, there are 2 centrioles that are present outside the nucleus of animal cell, and these act as basal bodies to make the flagella in some part of microvilli. They are part of MTOC. MTOC is microtubule organizing centers. Microtubules are very important. Microtubules are made up of a protein called tubulin. Now, tubulin is the protein that makes the cytoskeleton. That means the internal skeleton of the cell. And this is the protein that makes the spindle fibers during cell division. So centrioles and MTOCs are really important. When you look at the structure of tubulin, okay, tubulin has 2 shapes, alpha tubulin and beta tubulin. They combine together and they make the dimer, and there are 13 dimers which are arranged in a specific order. There is 25 nanometer gap between in between the lumen of this tubulin or microtubule, and again it is very important to make the cytoskeleton and to make the spindle fibers during cell division. Okay, then we have cilia and flagella. Again, flagella is present in some cells like bacteria. So in bacteria, their function is to propel the organism to move. It is very complex. It has 600 different types of polypeptides to control this movement of whipping movement specifically. And cilia are small hair-like structures which are also present in some of our cells, some of animal cells, and again on some microorganisms as well, like paramecium. They have cilia for their movement. When you look at the structure of bacteria, okay, these are electron micrographs. You can see the colony and you can see the singular cells as well. There are multiple flagella here. There are 2 flagella here. Singular is flagellum. There are pili here, and here you can see the colonies of bacteria. Now, the bacteria is considered to be very ancient, from which all the life has been developed. Their structure is much simpler as compared to eukaryotic cell. They do not have any membrane bounded organelle, no nucleus. DNA is randomly present in the cytoplasm. No histone proteins, no chromosomes, just the naked DNA. Ribosomes are 20 nanometer in size. They are 70S ribosomes, and they may sometimes have plasmids and pili, which are to connect with other bacteria to transfer the genetic material. And they may sometimes have capsule for extra protection. This is the diagram that gives you an idea. Now, this cell is much smaller than the eukaryotic cell. In volume, 1,000 times smaller. And in diameter, let's say the animal cell or plant cell could be around 40 to 50 micrometer in size, whereas the bacterial cell would be around 1 to 5 micrometer in size. So much smaller in diameter as well. And then we have the viruses. Bacteria are in the classification. They are considered as living organisms. They are living organisms. Viruses are not considered as living organisms. Because they are acellular, there is no cell. There is no cell membrane or cell wall or any of those structures that we have previously discussed. They only have 2 things. The outer layer, which is the capsid or the protein coat, and the inner core, which is made up of nucleic acid. Now, I am saying nucleic acid carefully because it could be DNA or RNA. Viruses are much smaller even than the bacteria. They are 20 to 300 nanometer in size. 1000 nanometer would mean 1 micrometer. Bacteria is larger than 1000 nanometer. They are 20 to 300 nanometer in size. They are all parasitic. Bacteria are not all parasitic. There are so many bacteria living in our body not causing any disease. But parasites are all, viruses are all parasitic. This is what the structure would look like under an electron microscope. This is the false color scanning. You can see the outer and inner structure. And that's all that we have in this chapter. Thank you very much. And I will keep you posted. So stay connected. I'll see you in the next video.